Okay, so welcome to this next video in which we're discussing type 2 activation of endothelial cells. Now, we're currently in the process of revising type 1 activation of endothelial cells, and we'll very soon get on to type 2 activation. Right, now I'm sorry for the interruption at the end of the previous video. So what we were doing was we were looking at neutrophil recruitment. This is the final portion of type 1 activation, okay? And this is very important in the endothelial cells, in the capillaries, and also the venules. So we've seen that in type 1 activation, you get the production of these vasorelaxant molecules, you get prostacyclin and nitric oxide being produced. And that's important in the arterioles, where it causes the vasorelaxation of the vascular smooth muscle cells, which then leads to vasodilatation of these arterioles, which increases blood supply to the affected area. We've then seen that you get endothelial contraction in the endothelial cells of the capillaries and the venules, where you get, which allows the formation of the inflammatory exudate in the infected area. And now what we're seeing is neutrophil recruitment, which is the movement of neutrophils from the bloodstream into the interstitial space. Okay, so let's say we have a neutrophil here, and I'm really upset now because I haven't left enough space, so I'm just going to have to, now I'm going to start this picture again. Right, so down here we'll have our endothelial cell, okay, and it's now undergone type 1 activation. So here is its nucleus. And because it's undergone type 1 activation, it's going to now express on its surface this P-selectin. So this is P-selectin. Now, neutrophils are a type of white blood cell. Okay? So let's draw a neutrophil here. And they're a type of white blood cell that has a multi-lobed nucleus. And the archetypal number of lobes for a neutrophil nucleus to have is 3. Okay, so this is a neutrophil. Neutrophils are also known as polymorphonuclear cells or polymorphonucleocytes, and for that reason they're often denoted PMNs, for polymorphonucleocytes. Okay, and that's because the number of lobes of the nucleus isn't set, and certainly if you look at these cells in histology, you'll see a varied number of lobes for the nucleus. Okay, so it can vary between 2 and you can see them with up to 5 generally. Okay, so on the surface of the neutrophil you have a protein known as PSGL1. Okay, and this stands for P-selectin glycoprotein ligand 1. P-selectin glycoprotein ligand 1. Okay, and this will bind to P-selectin on the surface of the type 1 activated endothelial cells. Okay, and when this happens, this is what's known as tethering of the neutrophil to the activated endothelial cell. Now, the endothelial cell also puts another molecule on the surface of its apical membrane. Okay, and I'm just going to colour in P-selectin vivid purple and P-selectin glycoprotein ligand 1 in blue. And this other molecule is known as platelet activating factor. Okay, so these endothelial cells are also going to put on their surface platelet activating factor, which is here. So PAF for short, for platelet activating factor. And basically the neutrophil also has a receptor for binding to platelet activating factor. So I'll put this here. Okay, so this is what's known as the PAF receptor, or the platelet activating factor receptor, which is often abbreviated just as the PAF-R. Okay, and when the PAF receptor binds to the PAF on the surface of the activated endothelial cell, this will cause a tight adhesion between the neutrophil and the endothelial cell. So the interaction between P-selectin and P-selectin glycoprotein ligand 1 forms the tethering. The interaction between the platelet activating factor and the platelet activating factor receptor forms a tight adhesion, and together they then produce the diapedesis of the neutrophils, where the neutrophils move across the endothelium. So let me draw this. Okay, right. So what will now happen is, if we draw our endothelial cells here, okay, then the endothelial cells, we know, have undergone this contraction process to open up a gap in between two neighbouring endothelial cells. Okay, here. And basically what's now going to happen is that you're going to sliver the neutrophil in between the endothelial cells. So it will sliver like a snake through this gap between the two neighbouring endothelial cells. So here's its trilobed nucleus again. Okay, and this process of slivering a neutrophil through the gap between the two neighbouring endothelial cells is what's known as diapedesis, or it's also called neutrophil extravasation. So neutrophil extravasation. Okay, and that's because you are taking the neutrophils into the extra vascular space, basically. Extravasation. You're taking them out of the vasculature. Right. Okay, so these then are the 4 things that are going to happen in type 1 activated endothelial cells. You're going to produce prostacyclin and nitric oxide, which will produce vasodilatation of the arterioles, increasing the blood supply and causing rubor and calor of the 5 pillars of inflammation. Then what happens is that you also get contraction of the endothelial cells. This is important in the capillaries and the venules, where it allows fluid and proteins to move out of the blood and into the interstitial fluid to form an inflammatory exudate, which is involved in attacking and also containing the infection. Okay, finally, you also start to recruit neutrophils to the site of infection, basically. So you take neutrophils from the lumen of the blood vessels and you move them via diapedesis out into the interstitial fluid. Neutrophils are then phagocytes, and they will go and attack the invading Staphylococcus aureus cells. Okay, right. So that's type 1 activation. Now, type 1 activation is extremely rapid. Activates in minutes, basically. The problem is that it also inactivates. Well, actually, it's part of the physiological response, so I shouldn't really say the problem. But the motivation for why we need another form of activation, namely type 2 activation, is that type 1 activation inactivates after around 10 to 20 minutes. So this process stops happening, or at least it goes down to very low levels after 10 to 20 minutes. Okay, now the reason for this is that the receptors that trigger type 1 activation are these GQ coupled 7 transmembrane receptors, okay, such as the H1 receptor. Now, if you stimulate these GQ G protein coupled receptors too often, then what will happen is they will be desensitized. So the cell will just stop responding to the ligand, basically. Now this could be because the receptors have actually been taken out of the membrane and sequestered into the endosome, or it could be that the G protein is just being uncoupled from the GQ pathway, so that even though the ligand still is binding to the receptor, it can't actually activate the pathway anymore. However, something happens that means that even though you're still throwing the ligand, which in this case is histamine, at the receptor, or at the cell at least, the cell no longer responds to it. So type 1 activation goes, basically. So we have another form of activation of endothelial cells, which lasts longer, basically. Okay, but it takes longer to initiate than type 1 activation because it involves changes in gene expression. Okay, so it's a longer time course that it takes. It takes hours to initiate type 2 activation, whereas it takes minutes to initiate type 1 activation. Okay, right, so now let's turn our attention to type 2 activation then. Right, so the starting point for the story in type 2 activation is that if we go back to our Staphylococcus aureus, which is infecting the tissue, okay, so here is our Staphylococcus aureus, growing like a bunch of grapes. Now, basically, these Staphylococcus aureus are detected. Basically, their presence does not go unnoticed. Just as we have the mast cells, there are other cells that are standing all around in every tissue of your body, waiting and watching. They are cells which are waiting and watching for an intruder. And these sort of cells are known as sentinel cells, which means that basically they stand on guard and are waiting for an intruder. So when the Staphylococcus aureus arrives and is starting to infect our tissue, these sentinel cells are going to notice it, basically. They're going to be the first to notice it. So you have two types of sentinel cells. Okay, so let me discuss the two main types of sentinel cells. And by the way, the mast cells that we discussed in type 1 activation, they are not a sentinel cell. They will be activated by sentinel cells. Okay, so the sentinel cells are the ones which first detect the problem. So here's our first type of sentinel cell. So it has this sort of star-shaped appearance, and it's what's known as a dendritic cell. Now these are not anything to do with neurons. So even though it may look like a neuron with dendrites coming off it, and it does, that's why it's called a dendritic cell, because it does have dendrites, it is not an excitable cell. It cannot conduct action potentials. Okay, so what this dendritic cell is a type of sentinel cell. There is another type of sentinel cell, and I'll discuss what they do after I've discussed the second type. So the second type is what's known as a resident macrophage. Okay, so this is a resident macrophage. So dotted around in all your tissues of your body, you have sentinel cells such as resident macrophages and dendritic cells. And these have receptors for molecules which are associated with non-self cells. So basically, these bacterial cells, these Staphylococcus aureus cells, and I'll label them up again, so they remember this this species of bacterium known as Staphylococcus aureus. At least we're suspecting that they are. So we've got this Staphylococcus aureus infection, okay, and basically the Staphylococcus aureus bacteria have all sorts of molecules which our cells, the eukaryotic cells of the body, would never have, okay? And these sort of molecules, which invading cells will possess, but normal cells of the body will not possess, have a name. They are generally referred to as pathogen-associated molecular patterns, okay? And that name makes sense because the word pathogen just means anything which is capable of causing pathology. So Staphylococcus aureus, this bacterium, certainly is a pathogen. It's capable of causing toxic shock syndrome. So it's certainly capable of causing pathology. So this is a pathogen, and it's got associated with it a bunch of molecules or a bunch of molecular patterns that our cells, the cells of the human body, would never ever use. Okay, so there are certain molecules that the Staphylococcus aureus has which would never ever be in a human cell. Okay, so they are a giveaway sign that this cell is not a normal human cell and that it's not supposed to be there. By the way, pathogen-associated molecular patterns are often abbreviated to PAMPs. Okay, now the sentinel cells, the resident macrophages and the dendritic cells, okay, these have in their cytoplasm and on their cell membrane receptors for these pathogen-associated molecular patterns. So they have proteins which will bind to and be activated by the presence of these molecules that are associated with pathogens. Okay, and these sort of receptors are generally referred to as pattern recognition receptors. So let me put these down here. So pattern recognition receptors. Okay, and these pattern recognition receptors, these will then activate the dendritic cell or the resident macrophage if they ever find their ligand. So if the Staphylococcus aureus is present, it will have all sorts of pathogen-associated molecular patterns associated with it, which will trigger the activation of these pattern recognition receptors on the surface of the sentinel cells. Okay, now when these pattern recognition receptors become activated on the sentinel cells, then they will lead to the activation of these sentinel cells, and the sentinel cells will start releasing what are known as pro-inflammatory cytokines. And this is one of those words that everyone hates, because people ask, well, what is a cytokine? And don't worry, I'll tell you exactly which molecules I mean in this situation. So they are known as pro-inflammatory cytokines. So a cytokine is just any molecule which signals between two cells. However, cytokines are usually associated with signaling in immunology, basically, immunological signaling between cells. Okay, so these pro-inflammatory cytokines are going to tell other cells that there is a major problem, basically, that there is pathogens within the tissue, basically, that there is this Staphylococcus aureus within the tissue. Okay, so the two main pro-inflammatory cytokines which we're going to study that these sentinel cells, dendritic cells and resident macrophages will release are called, the first one is called interleukin-1, and that's often abbreviated to IL-1. So this is a very famous molecule, interleukin-1, the first of the interleukins, and there are many interleukins. Okay, so I might even circle it with a colour because it's so important. So interleukin-1, which is getting its name circled with an orange box. Okay, and then the other pro-inflammatory cytokine which these sentinel cells release when activated by the presence of a pathogen-associated molecular pattern is tumour necrosis factor. And again, this is a very famous molecule. However, it's also often referred to as TNF-alpha. Now, there are different types of TNF, okay? However, the other types are pretty much trivial. So when people just say TNF or tumour necrosis factor, you can assume that they mean tumour necrosis factor alpha. Really, they should say specifically that they mean tumour necrosis factor alpha, but it's kind of like when people are talking about the GQ pathway, they'll often just talk about the alpha-Q GTP complex activating phospholipase C. Really, they should say specifically phospholipase C of the beta type, but the phospholipase C of the beta type is the most important type compared to the other types. And it's the type that's activated by the GQ pathway. So people often just do away with the specific isoform and say just phospholipase C. It's the same here, basically. Tumour necrosis factor alpha is the main form of tumour necrosis factor. There are other forms which aren't released by these sentinel cells. But if someone says just tumour necrosis factor without a clarification, i.e. without an alpha or a beta behind it, you can assume that they just mean tumour necrosis factor alpha. Okay, so this is tumour necrosis factor. Okay, and then I'll put the alpha there just to be correct. So it is tumour necrosis factor alpha. If you can remember to put the alpha, great, put it there. But you will often hear people refer to it just as tumour necrosis factor. So don't let that confuse you. So I would advise that you put the alpha to clarify it for people, but if you just read tumour necrosis factor, which you will in many places, assume that it means tumour necrosis factor alpha. Okay, right, so these sentinel cells, in response to the presence of the Staphylococcus aureus, have released these two pro-inflammatory cytokines, interleukin-1 and tumour necrosis factor alpha. Okay, and we'll see the role of these two in the next video, but basically they are going to activate, in the type 2 manner, endothelial cells.